Today is the 11th problem solving session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goel, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. Today we'll discuss the questions related to 11th week's topic. Today we'll be discussing about different processes like photosynthesis and all, and we'll study about different pigments involved in photosynthesis, and we'll discuss about certain diseases and the microorganisms associated with those diseases. So, I have certain questions related to these topics. We'll discuss the answers to those questions. If you have any doubt in any of the questions, please let me know. If you have any other doubt in any of these topics, do let me know. I'll help you to clarify the doubt. Okay. Sorry. So let's start with the questions. First question is ringworm is a disease caused by option A fungus, option B bacteria, option C tapeworm, and option D roundworm. Ringworm is a disease caused by option A fungus, option B bacteria, option C tapeworm, and option D roundworm. If you know the answer, write it in the chat box. Yeah, so you have written option A. Option A, that is fungus. That is the correct answer. So ringworm is caused by fungus. So ringworm, also known as dermatophytosis or dermatophyte infection, is a fungal infection of the skin. Ringworm infection can affect both humans and animals. Infection initially appears as red patches on the affected areas of the skin and later may spread to other parts of the body. It may affect the scalp, feet, nails, groin, beard, or other areas. Okay, so it is a fungal disease, it's a skin disease. So you must have seen ads for this ring guard cream. So that is actually for ringworm. So it causes a ring shaped uh, skin red patch on the skin. So it causes a ring shaped red patch. So that's why it's called a ringworm. And since it's a skin related disease, it's named as dermatophytosis or dermatophyte infection. Derma uh, is the term used for skin related stuff. Okay, so ringworm is caused by fungus. Moving on to the next question. Question number 2. Teardrops are rich in option A amylase, option B phosphate, option C lysozyme, and option D protease. Teardrops are rich in which enzyme? Amylase, phosphate, lysozyme, and protease. Okay, you have answered option C, lysozyme. Lysozyme, that is the correct answer. So teardrops are rich in lysozyme. So lysozyme is the enzyme that is found in the secretions of the lacrimal glands, that is the tears of animals, and in nasal mucus, gastric secretions, and egg white. So lysozyme is the enzyme that can break down the cell walls of certain bacteria. Okay. So if you remember, I told you the bacterial cell wall is composed of peptidoglycan, right? So it can actually break down the carbohydrate, that peptidoglycan basically, uh, that is found in the bacterial cell wall. And it is present in the secretions of animals to prevent it's basically a defense mechanism of our body. It prevents our body from getting bacterial infections. So, since lysozyme can break down the bacterial cell wall, any foreign bacteria, infectious bacteria, it cannot enter. So that's why it is present in the nasal mucus. So when you breathe in air, you are automatically going to breathe in certain bacterial cells, and certain bacterial cells can be infectious. So, to prevent uh, the infection from happening, the body has evolved in such a way so that it has certain mechanisms, certain compounds, certain enzymes to kill those bacteria. So, basically, it will break down the cell wall of the bacteria. Eventually, the bacteria will die and will not cause the infection in the body. Okay, that's clear.
Yes, ma'am.
Okay. Then moving on to the next question, question number 3. What is the importance of the epidermis regarding human immunity to microbes? Option A, it is the first layer of the skin which helps in defending the human body from microbes. Option B, epidermis keeps the body temperature sufficient to kill the microbes. Option C, there is no specific role for epidermis in preventing diseases. Option D, it is the last layer of the skin which helps in defending microbes from reaching the human body. So, what is the importance of epidermis regarding human immunity to microbes? It is the first layer of the skin which helps in defending human body from microbes, or it keeps the body temperature sufficient to kill the microbes, or is it no it or it has no specific role in preventing diseases, or it is the last layer of the skin which helps in defending microbes from reaching the human body. What do you think is the answer? You have answered option A. That is, it is the first layer of the skin which helps in defending the human body from microbes. That is the correct answer. So, human skin is made of three main layers. We have the epidermis at the outermost layer. Then we have the dermis and hypodermis. Okay, so there are three layers: epidermis, dermis, and hypodermis. So, epidermis is the topmost layer and it is like a covering for our body. So, whenever you buy a food from the market, it is generally covered in a plastic wrapper, right? So think of the epidermis as the wrapper that is protecting our body from the outside world. Okay, so if any food item is sold without any packaging and the same food item is sold with packaging, we generally prefer the one which is sold with packaging because we think it is more hygienic. Why do we think like that? We think like that because it is not coming in contact with the air, with the surrounding environment, so that we are more sure that it has less pathogenic bacteria growing on that as compared to the one that is not packaged. Similarly, if our body did not have this epidermis layer, our body would have been in contact with the outside world with all the pathogenic microbes directly. Okay, so epidermis is kind of that wrapper that is preventing our body from coming in contact with those pathogens directly. We do get infections anyway, but chances of us getting infection from these pathogenic microbes are highly reduced because of the epidermis, because of the topmost layer of our skin. Okay, I hope that's clear. I'll move on to the next question. Question number 4. What causes bubonic plague? What causes bubonic plague? Option A, Streptococcus mutans. Option B, Porphyromonas species. Option C, Prevotella intermedia. And option D, Yersinia pestis. What causes bubonic plague? Streptococcus mutans, Porphyromonas species, Prevotella intermedia, Yersinia pestis. You have answered option D, that is Yersinia pestis. Okay, let's see. That is the correct answer. Bubonic plague is caused by Yersinia pestis. So, Yersinia pestis is a bacterium. So, bubonic plague is one of the three types of plague caused by plague bacterium, that is the Yersinia pestis. Bubonic plague is an infection spread mostly to humans by infected fleas that travel on rodents. So, it's a flea, it's a type of an insect that generally is present on the rodents. So, oriental rat flea, that is Xenopsylla Cheopsis. Uh, so this is a flea that is present on the rodents. So here's a picture of that flea, of that insect. And this insect is generally infected by the plague bacterium, that is Yersinia pestis, which appears as a dark mass in the gut. So here you can see the Yersinia pestis. It basically resides in the gut of these insects, and the foregut of this flea is blocked by Yersinia pestis biofilm. So Yersinia pestis, what they do to survive inside these insects, they form a biofilm. Okay, to survive inside these insects, they form a biofilm. What is a biofilm? So biofilm is a structural form that is made by these bacterium. So a lot of bacteria can form biofilm as a defense mechanism, as to like in order to survive or in order to flourish themselves. They form these biofilms. So what they do, they attach their cells to each other and form a film like structure, a continuous film like structure, a continuous sheet like structure. And that when they do that, that basically makes it harder for any drug or for any agent to break down. Okay, if we have individual bacterial cells, it is easier for certain enzymes or certain medicines, certain drugs to break down those bacterial cells. But when they form biofilm, it gets more difficult for the chemical agent to attack the cells. Okay, so here also Yersinia pestis also forms the biofilm in order to survive in the gut of these insects. And since they form these uh, biofilms, they kind of block the foregut of the flea, of this flea. Okay, and when this flea attempts to feed on an uninfected host, Yersinia pestis from the foregut is regurgitated into the wound, causing the infection. So when this flea bites an uninfected host, so from that bite mark, from that wound that is caused because of the bite, uh, the bacteria, that is the Yersinia pestis, the plague bacterium, it enters from the insect to the host. Okay, so when this insect will bite an uninfected host, the bacteria that is present in the foregut, it will move from the bacteria from the insect to the host, and that's how the diseases spread uh, to humans. Okay, I hope that's clear. Moving
Yes,
on to
ma'am.
the next question. Next question, question number 5. Proteus species affect dash tissues and cause dash disease. Proteus species affect dash tissues and cause dash disease. So tissue option A is tissue is throat epithelium and disease is pyelonephritis. Option B is tissue is small intestine epithelium and disease is cholera. Option C is tissue is kidney medulla and disease is pyelonephritis. Option D, where tissue is kidney medulla and disease is spontaneous abortion in cattle. So Proteus species again, it's a bacteria. It affects which tissue and causes what disease? So you're saying B, that is small intestine epithelium and cholera. Okay, let's see the answer. The correct answer is not option B, it's actually option C. So Proteus species, it affects the kidney medulla and it causes pyelonephritis. So nephritis is a kidney disease. So it is caused by these Proteus species. Cholera is actually caused by another bacteria. So Proteus species, they infect these kidney medulla. So this is the medulla region of the kidney. So this is the healthy kidney and this is the one which is infected by these bacteria. So you can see inflammation is occurring in the kidney and then the kidney is filled with pus and slowly it's uh, stops basically it stops functioning eventually. So kidney infection, that is pyelonephritis, it's a type of urinary tract infection that generally begins in the urethra or bladder and travels to one or both the kidneys. Okay, so it is a type of a urinary tract infection, it's a type of UTI. It begins in the urethra or the urinary bladder and it travels to one or both the kidneys. It causes inflammation in the kidney medulla and eventually pus can fill in the kidney and kidney can stop functioning. Okay, do you understand this?
Yes, ma'am.
Great. Moving on to the next question. Question number 6. Which among the following is not a host to host epidemic? Which among the following is not a host to host epidemic? Option A, H1N1. Option B, hepatitis. Option C, influenza. Option D, COVID-19. Which among the following is not a host to host epidemic? Write your answers in the chat box. You have written option C. What about others? Anyone else wants to answer? Which among the following is not a host to host epidemic? H1N1, hepatitis, influenza, COVID-19. Do you understand what host to host epidemic means? So, host to host epidemic basically means it can spread from one host to another directly. Okay. So, if I have certain disease, uh, So I can tell you that COVID-19 is not the answer, because COVID-19 is a host to host epidemic. You know that if I have COVID, and uh, if like I can uh, basically the disease can spread from me to an uninfected host directly, okay, through the aerosol or through the air transmission or to the breath, okay. So which among the following three is not a host to host epidemic? It's not a host to host epidemic, okay. Okay, I have one more answer, option B, that is hepatitis. Yes, that is the correct answer. Hepatitis is not a host to host epidemic. The rest all of it, H1N1, you must have heard of H1N1 virus also. H1N1 uh, was also very widespread, and it was causing a lot of infections. And then influenza. So these are basically flu viruses, H1N1, influenza, COVID-19. So all of these are flu virus, so they can spread from one host to another directly, okay. But hepatitis is a inflammation of the liver caused by hepatitis A virus. Virus is primarily spread when an uninfected or unvaccinated person ingests the food or water that is contaminated with the feces of the infected person. So, hepatitis A cannot transmit from the infected host to the uninfected person directly. It can only travel through fecal contamination. So, the virus is actually excreted out of the body through feces, and if the water you are ingesting or food you are ingesting has fecal contamination, then the virus can enter your body through food, through your food passage, okay. So disease is closely associated with the unsafe water or food, inadequate sanitation, and proper personal hygiene. So that is why we are always advised to wash our hands while having food, and do not eat food from any outside source which is not hygienic enough. So that these kind of diseases can be caused in case hygiene is not maintained properly. Okay. So hepatitis A is not a host to host disease. Rest all of the diseases can be spread from one host to another directly. Moving on to the next question, question number 7. Identify the vector of Lyme disease. Option A, Aedes mosquitoes. Option B, Anopheles mosquito. Option C, black-legged ticks. And option D, cattles. Identify the vector of Lyme disease. Aedes mosquitoes, Anopheles mosquitoes, black-legged ticks, and cattles. Write your answers in the chat box. What is the vector of the Lyme disease? Aedes mosquitoes, Anopheles mosquitoes, black-legged ticks, and cattles. Write your answers in the chat box quickly. Okay, I have one answer. Says option D. cattle. What about others? Everybody write it the answers on the chat box. Okay, another answer is option B, Anopheles mosquitoes. Okay. Anyone else? Hmm, Sorry. Anyone else? So the correct answer actually is option C, black-legged ticks. So Lyme disease is the most common vector-borne disease in United States. Lyme disease is caused by the bacterium Borrelia burgdorferi and rarely Borrelia mayoni. It is transmitted to humans through the bite of an infected black-legged ticks. The most common sign of infection is expanding red rash, known as erythema migrans, that appears at the site of the tick bite about a week after it occurred. So these are small ticks. They can bite the humans and they can transmit these bacterium that can cause the Lyme disease. So bacterium is Borrelia burgdorferi. So Borrelia burgdorferi basically resides inside these ticks, and when the tick bites the human, the bacteria from the insect, from the tick, gets transferred to the humans through the site of bite, and red rash appears at the site of bite. Okay, so that is the Lyme disease. The other two options, Aedes mosquitoes and Anopheles mosquitoes, these are actually vectors of very common diseases. So you must be knowing which diseases are caused by mosquito bites. Anybody? Any guess? What could be the disease that is caused by Aedes or Anopheles? Yes, malaria, dengue. So Anopheles mosquito is the one that carries the malarial parasite, and Aedes mosquito is the one that carries the dengue virus. So bite of Aedes mosquito can cause dengue, even it can cause Zika. Okay, so these viruses are carried by these mosquitoes, and they when they bite the human host, they basically transmit those viruses from its own body to the human body. Okay, and that's how we get the dengue fever. And then same goes for Anopheles. It carries the malarial parasite, that is Plasmodium falciparum, and the malarial parasite has a whole life cycle. So half of it goes in the Anopheles mosquito and half of it goes in the human host. So when the mosquito bites the human host, it basically transmits these parasites, and then the parasite uh, grows inside our body and causes the malarial disease. Okay. I hope that's clear. Okay, moving on to the next question. Question number 8. Identify the non-zoonotic diseases from the following. Identify the non-zoonotic diseases from the following. Option A, anthrax. Option B, rabies. Option C, salmonellosis. Option D, malaria. Identify the non-zoonotic diseases from the following. Anthrax, rabies, salmonellosis, and malaria. Please write your answers in the chat box. Anybody? Which of the following is a non-zoonotic diseases? Anthrax, rabies, salmonellosis, or malaria? Okay, I have one answer. Option A, anthrax. Anyone else? No one knows the answer. Okay, let's see the correct answer. The correct answer is actually option A, anthrax. So you are right. So zoonotic disease basically means the diseases that are transmitted from animals to humans. Okay. So a zoonosis or zoonotic disease or zoonosis as plural. It is an infectious disease that is transmitted between species from animals to humans or from humans to animals. And anthrax is a serious infectious disease caused by gram-positive rod-shaped bacteria called Bacillus anthracis. Anthrax can be found naturally in soil and commonly affects domestic and wild animals around the world. Inhalation anthrax can occur when a person inhales the spores that are present in air that are aerosolized during the industrial process of contaminated materials such as wool, hides, or hair. Okay, so Bacillus anthracis causes anthrax. The spores of these bacteria can be present in the air as aerosol formation, and when we inhale these spores, we can suffer from anthrax. Okay, so this disease is not caused from an animal. Other than that, the other options, rabies, salmonellosis, and malaria, all of them are actually transmitted from animals to humans. So rabies, you must be familiar with this. So most of you have heard of this disease, I hope. So it's a disease that is caused by a virus. So this virus mainly is transmitted through direct contact, such as through broken skin or mucous membranes in the eyes, nose, or mouth, with saliva or brain nervous system tissue from an infected animal. So, generally, you must have heard of rabid dogs. Okay, so generally, these viruses they infect these animals. So, dogs, cats, and all other domesticated animals, and even the wild animals. The virus can infect those animals, and when the animal bites a human being, the virus can be transmitted from the saliva of those animals through the wound of the human. Since they are biting, they are breaking our skin, and the virus can enter our body through that broken skin. Okay, so that's how we can get rabies, and this basically affects the brain or the nervous system of our body. So that's why it is also said that when a rabid animal bites the human, so the site of bite is very important, the location where it is biting. So if a rabid animal bites me in my leg, uh, the time for the virus from so the virus is going to enter from the leg because it is bite mark is there, but the time for the virus to travel from my leg till my brain will be longer as compared to a situation where a rabid animal bites me on my head. Okay, so if a rabid animal bites me on my head, chances of me getting infected by the rabies uh, virus and my nervous system or my brain can get infected by this virus is very, very high as compared to the case where my leg has been bitten. So the location of the bite is very important in case of rabies because the virus has to travel to the nervous system. Okay, and that is why as soon as you get a uh, like you get bitten by a rabid animal, you should get the uh, injections that are basically there to prevent the rabies infection. Okay. Next is salmonellosis. So, salmonellosis is caused by salmonella. It's again a group of bacteria that can cause gastrointestinal illness and fever, which is salmonellosis. And it can spread from animal to people. People who have direct contact with certain animals, including poultry and reptiles, can spread the bacteria from animals to the food if they do not practice proper hand washing hygiene before handling the food. So these bacteria can spread from animals to humans. That's why these are also zoonotic diseases. So generally, salmonella, the name comes from salmon. Salmon is a type of fish, and the bacteria was found there for the first time, and these bacteria generally resides in poultry and fish and reptiles. So if you are eating uh, chicken or fish, and if you don't wash it properly, if you don't clean it properly, or if you don't cook it properly, these bacteria can be present. So proper cooking of food is important for prevention of bacteria of these kind of infections. Other than that, if the person who is handling poultry uh, is not washing his hands properly and he's eating his food, he can get the salmonella infection. Okay, and It causes gastrointestinal illness. So basically, uh, stomach issues will be happening. And the next was malaria. I have already talked about malaria. So it's a mosquito-borne disease. I told you, Anopheles, female Anopheles mosquitoes are the ones that carry these parasites. So, malaria causes symptoms that typically include fever, tiredness, vomiting, headaches, and in severe cases, it can cause yellow skin, seizures, coma, or death. Okay. Moving on to the next question. Question number 9. Sandflies spread the deadly disease which is known in India as Kala Azar. Which microbe causes this disease? Option A, Leishmania protozoan. Option B, uh, Ike virus. Option C, Yersinia pestis bacteria. And option D, Plasmodium parasites. What would be the correct answer? Write your answers in the chat box. Sandflies spread the deadly disease which is known in India as Kala Azar. Which microbe causes this disease? Okay, you have written option A as the answer. Anyone else? Hmm. Anyone else wants to answer? So option A is the correct answer, Leishmania protozoan. So visceral leishmaniasis, known as Kala Azar, is fatal if left untreated in over 95% of cases. It is characterized by irregular bouts of fever, weight loss, enlargement of spleen and liver, and anemia. Most cases occur in Brazil, East Africa, and India. Leishmaniasis is caused by protozoa parasite from over 20 Leishmania species. Over 90 sandfly species are known to transmit the Leishmania parasites. Okay, so it's a protozoan, Leishmania, which causes this disease Kala Azar. It is commonly known as Kala Azar. It causes irregular bouts of fever, weight loss, and symptoms like that. Okay. Moving on to the next question, question number 10. Amoebiasis is a waterborne disease. What part of human body is mostly affected by this parasitic disease? Option A, eyes. Option B, ears. Option C, urinary tract. And option D, colon. Eyes, ears, urinary tract, or colon. Amoebiasis is a waterborne disease. What part of the human body is mostly affected by this parasitic disease? Okay, I have one answer, option D. What about others? All of you write your answers in the chat box. Anybody else? Come on, all of you try to answer. What part of human body is affected by this parasitic disease? Amoebiasis. Eyes, ears, urinary tract, or colon? Okay, let's see the answer. Option D is the correct answer, colon. So, amoebiasis is a parasitic infection of the colon or intestines caused by the protozoan Entamoeba histolytica. So, this protozoa causes this disease. Symptoms of amoebiasis includes loose stool, abdominal cramping, and stomach pain. However, most people with amoebiasis won't experience significant symptoms. In some cases, it invades the colon wall, causing colitis, acute dysentery, or long-term diarrhea, that is chronic. Infection can also spread through the bloodstream into the liver. Okay, so in some cases, it can become actually dangerous, but most of the time, it's like normal diarrhea, diarrhea-like symptoms. Okay, so Entamoeba histolytica causes amoebiasis, and it affects the colon of the human. Okay, I hope that's clear. If anybody has any doubt in any of the questions discussed till now, please ask. Okay, moving on to the next question. Question number 11. The relative ability of a parasite to cause disease is called option A vector, option B virulence, option C virality, and option D viral ability. Relative ability of a parasite to cause disease is called vector, virulence, virality, or viral ability. Okay, Smita says option B, that is virulence. What about others? Anyone else wants to answer? Please write your answers in the chat box. Hmm, Sorry. Okay, I have another option. Option A. Okay. Relative ability of a parasite to cause disease is called. One of you says vector, one of you says virulence. Let's see the answer. The correct answer is option B, virulence. So virulence is a pathogen's or microorganisms' ability to cause damage to a host. Okay, so any microorganism, if it is able to cause a disease, a- it's called a virulent organism. Okay, and the relative ability basically is measured by the virulence. So, uh, one can be more virulent and one microorganism can be less virulent. So, it can be used as a relative ability. It can be used as a comparison term. Okay. Vector is basically the, uh, how do I say? Vector is basically the agent that is transmitting the disease from one host to another. So, like in case of malaria, it is caused by parasite, right? It is caused by plasmodium. But mosquito will be the vector. Mosquitoes, they are transmitting that parasite from one host to another. Okay, one human being to another. So that is the vector. The one that is transmitting the disease, that is known as the vector. Okay. So, in case of malaria, dengue, mosquitoes are the vector. Okay. In case of, what did we discuss? Uh, bubonic plague, we were discussing, which was caused by Yersinia pestis. So, Yersinia pestis was transmitted by uh, a rat flea, right? So, basically, whatever insect what is carrying the uh, microorganism, that is called the vector. Okay, and virulence is the ability to cause damage to the host. The ability of a microorganism to cause damage to the host. Is that clear?
Yeah, ma'am.
Okay. Okay, moving on to the next question then. Question number 12. Microbe human interaction can be neutral, mutually beneficial, or pathogenic. In the following list of microbes, identify the mutually beneficial microbe out of the pathogenic ones. Okay. So microbe human interaction, it can either be neutral or it can be mutually beneficial. Mutually beneficial means it benefits both the microbe and the humans. Okay. Or it can be pathogenic, means the microbe can cause disease to the human. So, in the following list of microbes, identify the mutually beneficial microbe out of the pathogenic ones. So, out of these four, three are pathogenic, one is mutually beneficial. So, option A, Escherichia coli, option B, Mycobacterium tuberculosis, option C, Clostridium botulinum, and option D, Staphylococcus aureus. So, out of these four, three are pathogenic. Only one is mutually beneficial. So, which one of the following is mutually beneficial? Option A, Escherichia coli. Option B, Mycobacterium tuberculosis. Option C, Clostridium botulinum. And option D, Staphylococcus aureus. Okay. I have one answer, says option A, that is Escherichia coli. Okay. What about others? Everybody write your answers. Anyone else wants to answer? Nobody? Okay, so I have one answer, option A. Let's see if that's the correct answer or not. That is the correct answer. So E. coli, or that is Escherichia coli, it's a type of bacteria that normally lives in the intestines of all warm-blooded animals. Except E. coli, all other microbes that are given in the options are pathogens. So E. coli, it is normally found in the gut, found as the gut bacteria. So it helps maintaining a good gut health. Mycobacterium tuberculosis, it is the causative agent of tuberculosis. Uh, it's a disease that is caused in the lung. Clostridium botulinum, it forms a toxin called botulinum toxin, and it can cause paralysis to the uh, muscles. So I have talked about this in the previous sessions also. So this botulinum toxin, it is actually used as uh, a clinical as a cosmetological treatment agent. So Botox, you must have heard of it. So people use these toxins in very small quantity, these Botox, since it has the ability to cause paralysis to the muscles in very small quantity. When injected in the skin, it will cause paralysis of the skin, and it will prevent the skin from wrinkling. Okay, and that's how people prevent like people who want to look young, who want to prevent aging, they generally take these toxins. Okay, but in larger quantities, these toxins can cause muscle paralysis. Then Staphylococcus aureus, it's a very common bacteria. It is generally present in the skin, present on the skin. It generally causes these pimples on the face. So it's also a pathogenic bacteria. Okay. So all the other three bacterium are pathogenic. Only E. coli is the one that is mutually beneficial microbe. E. coli helps maintain a good gut health. It helps uh, in the food digestion process. Okay. Moving on to the next question, question number 13. Organisms that can make their own energy using light and carbon dioxide by the process of photosynthesis are option A, photoautotrophs, option B, chemoautotrophs, option C, photoheterotrophs, and option D, chemoheterotrophs. Organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis are called option A, photoautotrophs, option B, chemoautotrophs, option C, photoheterotrophs, option D, chemoheterotrophs. Write your answers in the chat box. Okay, I have only one answer. Option A, photoautotrophs. What about others? Everybody write your answers in the chat box. Hmm. Anyone else? Okay, so let's see the answer. The correct answer is option A, photoautotrophs. So you are right. Photoautotrophs are the organism that can make their own energy using light and carbon dioxide by the process of photosynthesis. So this is how the division of all the organism is done. So if the organism is able to use carbon dioxide as the carbon source, they are called autotrophs. If they are using organic carbon as the carbon source, they are called heterotrophs. Okay. Then comes the energy source. So if they are using light energy, then photo is used as prefix. So photoautotrophs or photoheterotrophs. If they are using chemical energy, then chemoautotrophs or chemoheterotrophs. Okay. So this is the basic division: photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Then in photoautotrophs, if uh, they use H2O to reduce the carbon dioxide, in that case, oxygenic photosynthesis will occur. In the absence of oxygen, anoxygenic photosynthesis will occur. So there are certain bacteria, green and purple bacteria, which can uh, do photosynthesis in the absence of oxygen. So other elements are there, sulfur or iron. These are generally then used. So in case of chemoautotrophs also, hydrogen, iron, sulfur, nitrogen, and carbon monoxide oxidizing bacteria are present. Then photoheterotrophs, green and purple non-sulfur bacteria. Then in chemoheterotrophs, again, if the terminal electron acceptor is oxygen, then aerobic respiration will happen, which is common in all the animals. We all do aerobic respiration, right? But if the terminal electron acceptor is not oxygen, in that case, there can be two process. One can be organic compound fermentative bacteria, that is lactobacillus is an example, or second is inorganic compound anaerobic respiration, like in Clostridium. So terminal electron acceptor, it can eat, if not oxygen, it can be sulfur or iron. So whatever is the terminal electron acceptor, based on that, that kind of respiration will be there. Okay, so if it is oxygen, it will be aerobic. If it is not oxygen, it will be anaerobic respiration. Okay, is that clear? Okay, moving on to the next question. The next question is based on the table that I've just explained. So everybody should be able to answer this if that is clear. If you're not answering this, then I would understand that you haven't understood the previous slide. Organisms that are able to synthesize their own organic molecules from the fixation of carbon dioxide and are able to produce their own source of food or energy are called. So they are able to synthesize their own organic molecule from the fixation of carbon dioxide and are able to produce their own source of food and energy. Option A, photoautotrophs. Option B, chemoautotrophs. Option C, photoheterotrophs. Option D, chemoheterotrophs. I have only one answer in the chat box. Everybody write your answers in the chat box. Everybody should be able to answer this. Nobody wants to answer? Okay, if Smita, you're saying option C, that is photoheterotroph. Uh, I've told you the whether the energy source is light or chemical. Based on that, we decide whether it is photo or chemo, right? And if they are producing their own food, then it would be autotrophs. If they are producing their uh, food using organic carbon source, then it will be heterotrophs. If they are using just carbon dioxide for making food, then it would be autotrophs. If they are using organic carbon source, then it would be heterotrophs. Now think again and write your answer. What do you think is happening in this question? Organisms that are able to synthesize their own organic molecules from the fixation of carbon dioxide and are able to produce their own source of food or energy are. Do you still think it's option C? Okay. Now you are saying option D. Okay. The correct answer actually is option B. It's chemoautotroph. So I told you if they are producing their own source of food, it is autotroph. So in this figure, I told you if it is using CO2 as the carbon source, that means they are making their own food, then it is autotroph. If they're using organic carbon, that means they are going to break down the organic carbon eventually to carbon dioxide. So they are heterotroph. Okay. So in this question, the organisms are able to synthesize their own organic molecule from the fixation of carbon dioxide. That means they are autotrophs. Okay. And they are able to produce their own food, own source of food and energy. So since they are not using light as the energy source, since they are not using light as the energy source, that's when they are not photoautotrophs. They are chemoautotrophs because they are obtaining their energy from a chemical reaction, and their source of carbon is most oxidized form of carbon, that is carbon dioxide. Okay, so here you can see if source of carbon is carbon dioxide, then it is autotroph. If the source of carbon is organic carbon, then it is heterotroph. If they're using light energy, then it is photo. If they're using chemical energy, then it is chemo. So in the previous question, we had the organism that can make their own energy using light. That's why it was photo, and carbon dioxide. That was it was autotroph. So photo autotroph. In this question, it is chemoautotroph. Okay, is that clear to you now?
Yeah, ma'am.
Okay. Clear to everybody? Anybody has any doubt in this? Moving on to the next question. What is the maximum absorption range of bacteriochlorophyll A? What is the maximum absorption range of bacteriochlorophyll A? Option A between 600 to 725 nanometer. Option B between 625 to 700 nanometer. Option C between 800 to 925 nanometer. And option D between 825 to 900 nanometer. What is the maximum absorption range of bacteriochlorophyll A? Between 600 to 725 nanometer, between 625 to 700 nanometer, between 600 to 925 nanometer, or between 825 to 900 nanometer. Write your answers in the chat box. Come on, everybody, write your answers in the chat box. What is the maximum absorption range of bacteriochlorophyll A? Between 600 to 725 nanometer, 625 to 700 nanometer, 800 to 925 nanometer, 825 to 900 nanometer. What is the correct answer? So the correct answer is option C, between 800 to 925 nanometer. So bacteriochlorophyll A is a pigment that can absorb in the range of 800 to 925 nanometer, which is kind of beyond the visible range spectra. So it can absorb the light of wavelength 800 to 925 nanometer. Okay. Moving on to the next question, question number 16. Membrane-bound pigments that participate in photosynthesis and photoprotection of the organisms are called. Membrane-bound pigments that participate in photosynthesis and photoprotection for the organisms are called. Option A, carotenoids. Option B, carcinogens. Option C, phycobilins. Option D, phytonadione. Option A, carotenoids. Option B, carcinogens. Option C, phycobilins. And option D, phytonadienes. Membrane-bound pigment that participate in photosynthesis and photoprotection for the organisms are called. You are saying option C, phycobilins. Okay. Anyone else? Anyone else wants to answer? Sorry. Anybody else wants to answer? So the correct answer is option A, carotenoids. So carotenoids are class of more than 750 naturally occurring pigments synthesized by plants, algae, and photosynthetic bacteria. These richly colored molecules are sources of yellow, orange, and red colors of many plants. Carotenoids are ubiquitous and essential pigments in photosynthesis. They absorb in the blue-green region of the solar spectrum and transfer the absorbed energy to bacteriochlorophylls, and so expand the wavelength range of light that is able to drive photosynthesis. So basically, these are pigments that are present in the plants along with chlorophyll. So chlorophyll is the major pigment that is present in the plants that is responsible for photosynthesis, right? So you must have read about chlorophyll and photosynthesis in your schools. So chlorophyll is the green colored compound, green colored pigment that is present in the plants that gives the plants its green color, and it helps in photosynthesis. Along with chlorophyll, the plant has certain other pigments. So you must have seen there are leaves that are red in color. That is actually because of the presence of the pigments like carotenoids, which can give yellow, orange, or red colors to many plants. So these pigments are present in all the plants in smaller or the larger quantity. So if it is present in larger quantity, it makes the plant appear reddish in color. And these pigments are present basically to widen the range of wavelength of light plants can absorb. So or even in case of photosynthetic microorganisms, algae and other photosynthetic bacteria, these pigments are present so that when the bacteria, let's say, is growing in a area where the light that is absorbed by chlorophyll is not present. Okay, in that case, the bacteria will evolve eventually to have a pigment that can absorb the light of the particular wavelength that is present in that particular region. So for that, in that case, carotenoids will be the pigment that can absorb the that can absorb the wavelength range present in that area. So it basically expands the wavelength range of light that is able to drive photosynthesis so that a organism can survive. Okay, is that clear? Is that clear to everybody? If you have any doubt in any of the questions discussed till now, please ask. Okay, moving on to the next question. Question number 17. Water soluble pigments not attached to the membranes are called. So carotenoids were membrane bound pigments. If you, I think yeah. So in the previous question, we had membrane bound pigments that participate in photosynthesis. So carotenoids were membrane bound pigments. Okay. Now in this question, you have to answer which of the following is a water soluble pigment that is not attached to membrane. Okay, option A, carotenoids, option B, carcinogens, option C, phycobilins, and option D, phytonadione. Okay, so if Smita says option C, phycobilins, anyone else? Come on, everybody, write your answers in the chat box. Only one person is answering. Water soluble pigments not attached to membranes are known as option A carotenoids, option B carcinogens, option C phycobilins, and option D phytonadione. Okay, let's see the answer. The correct answer is option C, phycobilins. So you are right. So phycobilins are the light capturing bilins found in cyanobacteria and in the chloroplasts of red algae, glaucophytes, and some cryptomonads, though not in green algae and plants. Um, Most of their molecules consist of a chromophore, which makes them colored. So phycobilin also gives a reddish brownish color. Okay, so it has a chromophore. Chromophore is a structure which can give color to a compound. Okay, so generally it's an aromatic structure which can absorb a particular wavelength of light, giving uh, a particular color. Okay, so they are unique among the photosynthetic pigments in that they are bonded to certain water soluble proteins known as phycobilin proteins. So that's why these are water soluble pigments. Phycobilin proteins then pass the light energy to chlorophylls for photosynthesis. Phycobilins are especially efficient at absorbing red, orange, yellow, and green light wavelengths that are not well absorbed by chlorophyll A. So I told you, along with chlorophyll, multiple other pigments are present in these photosynthetic organisms because chlorophyll cannot absorb a wide range of light wavelengths. So basically, these other pigments they help absorbing in other wavelengths. So chlorophyll A can absorb in around 600 and 400 nanometers of wavelength. So you can see phycobilins can absorb in the red, orange, yellow, and green light wavelength range. So that means it can widen the absorbance range of light for any organism. So organisms growing in shallow waters tend to contain phycobilins that can capture yellow or red light, while those at greater depths often contain more of phycobilins that can capture green light. Okay, so there are multiple types of phycobilins. So the organisms that are growing at in the shallow water, they can get direct sunlight. Okay, some amount of sunlight can penetrate the water, right? So that's why in those organisms phycobilins that can capture red or yellow light is present. Okay, but the ones that are present deep inside the water, there the light is not uh, going to penetrate that much. So green light, uh, the ones that can absorb green light, the phycobilins that can absorb green light, that is present in the organisms that grow in the greater depth. Okay, because green light is more abundant there. Phycobilins fluoresce at a particular wavelength and are therefore often used in research as a chemical tag. Example, by binding phycobilin proteins to antibodies is a technique known as immunofluorescence. So, since these have a chromophore, these phycobilins have a chromophore. So these chromophoric structures, they can basically absorb the light of a particular wavelength and emit it at a different wavelength, giving them that color. So because they can absorb a light at a particular wavelength and then emit, it is known as a fluorescent molecule. And since it can give fluorescence, it is generally used as a chemical tag for certain studies, for immunofluorescence studies. Okay, is that clear to everybody? Any doubt? Okay, moving on to the next question. Number of electrons required for the nitrification of ammonium is option A 4, option B 6, option C 8, option D 10. Number of electrons required for the nitrification of ammonia. What is nitrification of ammonia? Basically conversion of ammonia or ammonium into nitrite and then nitrate. Okay, from ammonium, from NH4 plus, it will go to NO2 minus and then NO3 minus. So how many electrons will the ammonium ion require to become nitrate, final nitrate? Okay, option A 4, option B 6, option C 8 and option D 10. Write your answers in the chat box quickly. Nobody? Nobody knows the answer? Okay, let's see the answer. The correct answer is option C. So nitrification is the process by which ammonia and ammonium is converted to nitrate, as I've already mentioned. Nitrification is the net result of two distinct processes. So first step is ammonium to nitrite, that is NH4 plus to NO2 minus, by nitrosifying or ammonia oxidizing bacteria, and then further oxidation of this nitrite, NO2 minus, to nitrate, that is NO3 minus, by nitrite oxidizing bacteria. Okay, first ammonium oxidizing bacteria and then nitrite oxidizing bacteria. So these are the steps. So first the ammonium ion is converted into nitrite, right? Ammonium ion is oxidized into nitrite. So how do you find the number of electrons? You have to balance this equation. So you have one nitrogen this side, one nitrogen this side. So nitrogen is already balanced. Let's balance the oxygen first. So you have two oxygen here. So how do we balance oxygen? By adding water molecules. Okay, so if we add a water molecule on the left hand side, two water molecules on the left hand side, the oxygen will be balanced. Now balance the hydrogen. So how many hydrogens are there? We have 4 hydrogens here and then 2 to the 4. So 4 and 4, 8 hydrogens are there. So we will add 8 protons on the right hand side. Okay. So now we have the equation NH4 plus 2H2O goes to NO2 minus plus 8H plus, right? Now you see the charge. So you have plus 1 charge here and you have 8 minus 1, that is plus 7 charge here. So you have to balance the charge also. Okay. So you have to balance the charge now. So first you balance the oxygen, then you balance the hydrogen, then you balance the charge. So charge, here you have plus 1, here you have plus 7. So if you add 6 electrons on the right hand side, this will be also plus 1. So this is minus 6, this is minus 1. So minus 7 plus 8. So that means plus 1. And here also plus 1. So that means this reaction is requiring 6 electrons for ammonium to get oxidized into nitrite, right? So this is just the first step of ammonification. Now, second step, what is the second step? Second step is the conversion of nitrite to nitrate. So NO2 minus goes to NO3 minus. So again, we'll balance the oxygen first. So we have 2 oxygen on this side and 3 oxygen on this side. So we'll add the water molecule for adding 1 extra oxygen. Then we'll balance the hydrogen. So we have 2 hydrogen. So we'll add 2 protons here. Then we'll balance the charge. So we have -1 charge here and we have 2 minus 1, that is +1 charge here. So if we add 2 electrons, here also we will have -1 charge, right? So -1 +2 -2. So that means total charge is -1. Okay, so 2 electrons are required for the oxidation of nitrite to nitrate. Okay, so 6 electrons for the oxidation of ammonium to nitrite and then 2 electrons for nitrite to nitrate. That means total 8 electrons are required for conversion of ammonium to nitrate. Okay, so nitrification of ammonia, that is ammonium to nitrate, the whole process requires 8 electrons. Okay, is that clear? Anyone has any question? If anyone has any doubt, please ask. I hope everybody understood how to calculate the electrons. Okay. Moving on to the next question, question number 19. The order of zones in a Vinogradsky column is option A, aerobic water, anaerobic sediment, and anaerobic water. Option B, aerobic water, anaerobic water, then anaerobic sediment. Option C, anaerobic sediment, aerobic water, anaerobic water. Option D, anaerobic water, aerobic water, then anaerobic sediment. Top to bottom, you have to tell the zones that will be present in a Vinogradsky column. Okay, from top to bottom, whether it will be option A, aerobic water, anaerobic sediment, anaerobic water. Option B, aerobic water, anaerobic water, anaerobic sediment. Option C, anaerobic sediment, aerobic water, anaerobic water. Option D, anaerobic water, aerobic water, anaerobic sediment. What would be the answer? Write your answers in the chat box. I have talked about Vinogradsky column when we were discussing the history of microbiology. So in a Vinogradsky column, it basically is a system that mimics the uh, pond ecosystem or any water body. Okay, so think about that. Think about a lake ecosystem or a pond ecosystem and think whether the aerobic water would be on the top or anaerobic water would be on the top or anaerobic sediment would be on the top. Then what would come next? Then what would come at the bottommost layer? If Smita says option C. Okay. Anyone else? Anybody else wants to answer? Okay. So the correct answer is option B. Aerobic water would be at the topmost layer, then anaerobic water, and then anaerobic sediment. So this is how a Vinogradsky column looks like. So I told you it is experimental setup to mimic the pond ecosystem. So basically so this is a common experiment that we also perform in microbiology course. So what we do is we take a long measuring cylinder, 2 liter measuring cylinder, and we put soil at the bottom of the column, and then we fill it with water. Okay. So, and we add certain nutrient, certain carbon source. So you can add newspaper, uh, shredded newspaper as a carbon source. You can add some nitrogen source and sulfur source. Or you can take the soil containing all these nutrients, okay? And then you have filled it with water. So what will happen in that column, and you keep the column in the sunlight, okay? So what will happen? Light will penetrate up to a certain extent in the column, and then oxygen will penetrate up to a certain extent in column. So oxygen can only penetrate at the upper surface layer. So that part will be aerobic water where the oxygen is diffusing through into the water. After that layer, there will be no oxygen. So that water would be anaerobic water. And then at the bottom, the sediment part, the soil part, that will form the anaerobic sediment. So in any pond or lake ecosystem, it has a bed which is at the which is the bottommost part of the lake or the pond. It is the bed of the bed of the lake or bed of the river, right? So that part is more anaerobic, no, because oxygen is not going to penetrate that. That part is going to have no light also. Then you will have the water just above that layer would be anaerobic water, and then at the topmost part you will have the aerobic water. So in the aerobic zone, you will have the chemoautotrophs and cyanobacteria that are photoautotrophs. So you will have all the uh, oxygenic photosynthetic organism. Then you will have sulfur oxidizing bacteria. Anoxygenic photosynthesis will occur. Colorless bacteria that uses H2S as the energy source. Then aerobic chemoheterotrophs. In the anaerobic zone, you will have photoheterotrophs, purple non-sulfur bacteria, which will use light as the energy source and organic carbon as the carbon source. Then anaerobic photosynthetic purple and green sulfur bacteria, which will do anoxygenic photosynthesis, that is anaerobic photosynthesis without oxygen. Then below that, we'll have anaerobic respiration. Then below that, we'll have the sulfur reducing bacteria. Sulfur and sulfates are reduced. And then at the bottommost layer, you will have the heterotrophic fermentation where the sulfates are reduced to sulfides. Okay. And these are some examples of the bacterium that are present in the column. So when you perform this experiment, when you fill it with soil and all the nutrients and fill it with water completely till the brim, these bacteria will slowly start growing and divide themselves based on the areas where they can survive. Okay, so this is a classic experiment performed by Vinogradsky to mimic an ecosystem and to study the biodiversity in one column inside a lab. Okay, is this clear? Do you understand the Winogradsky column? Please ask if you have any doubt. Okay, great. Moving on to the next question then, question number 20. Photosynthesis occurs in option A, chloroplast, option B, Golgi body, option C, endoplasmic reticulum, and option D, nucleus. Where does photosynthesis occur? Chloroplast, Golgi body, endoplasmic reticulum, or nucleus? Yes, you are saying option A, that is chloroplast. That is the correct answer. This is a very simple question. So photosynthesis occurs in chloroplast. Okay. Moving on to the next question. Question number 21. Final product of Calvin cycle is option A, RUBP, option B, glucose, option C, dihydroxyacetone phosphate, and option D, glyceraldehyde 3-phosphate. Final product of Calvin cycle is RUBP, glucose, dihydroxyacetone phosphate, or glyceraldehyde 3-phosphate. Okay, Smita says option B. Anyone else? Anyone else wants to answer? Please write the answer in the chat box. So the correct answer is option D, glyceraldehyde 3-phosphate. So Calvin cycle basically, here we have shown the cycle. So the diagram shows three turns of the cycle in which three molecules of carbon dioxide are fixed and one molecule of glyceraldehyde 3-phosphate is produced. So three times carbon dioxide will enter and it will basically form uh, glyceraldehyde 3-phosphate. So one time carbon dioxide will enter, again second cycle carbon dioxide will enter and go around, and then third cycle carbon dioxide will enter, and then we'll have the output of glyceraldehyde 3-phosphate. Okay, and two molecules of glyceraldehyde 3-phosphate will make one molecule of glucose. Okay, so two molecules of glyceraldehyde 3-phosphate are needed to make one molecule of glucose. Therefore, the cycle must turn 6 times for each glucose molecule. So three times cycle will turn to make one glyceraldehyde 3-phosphate. 6 times it will make two glyceraldehyde 3-phosphate, so that it can make one glucose molecule, produced requiring a total investment of 6 molecules of carbon dioxide, 18 molecules of ATP, and 12 molecules of NADPH. So this is a process that happens in plants where the carbon dioxide fixation occurs. That happens in all the autotrophs basically. Okay, so you remember you have studied about glycolysis in the previous session, right? Where glucose was the carbon source and it was broken down into carbon dioxide. So it is kind of the opposite process where carbon dioxide is fixed. So by autotrophs, autotrophs are fixing carbon dioxide, right? They are using carbon dioxide as the primary carbon source. So what they are doing, they are making glucose, and in that process, they are generating ATP. ATP is the major energy source. So whichever biological process is going on, biochemical process is going on, the final goal of any process would be to produce ATP. Okay, so when the glycolysis and TCA and electron transfer chain was happening in case of aerobic respiration, the final aim was to produce ATP. Here also for autotrophs, when they are using carbon dioxide as the carbon source, then also their main aim is to produce ATP. Okay, so You can see three molecules of carbon dioxide are used to produce one molecule of glyceraldehyde 3. Is that clear? Moving on to the next question. Reduction of sulfates and sulfides to hydrogen sulfide is done by which group of bacteria? Option A, aerobic sulfate reducing bacteria. Option B, photosynthetic sulfur bacteria. Option C, anaerobic sulfate reducing bacteria. And option D, heterotrophic bacteria. Reduction of sulfates and sulfides to hydrogen sulfide is done by which group of bacteria? So I told you in the Vinogradsky column at the bottommost layer, the sulfates and sulfides are converted into sulfides. So it is done by which group of bacteria? Aerobic sulfate reducing bacteria, photosynthetic sulfur bacteria, anaerobic sulfate reducing bacteria, or heterotrophic bacteria. What would be the correct answer? I've given you the hint. It is happening at the bottommost layer of the Vinogradsky column. So which organism would it be? I've just explained you the Vinogradsky column. Tell me which organism grows at the bottommost layer of the column. What would be the answer to this question? Come on, write your answers in the chat box. If Smita, you are saying photosynthetic sulfur bacteria, do you think light can penetrate to the bottommost layer of the Vinogradsky column? For photosynthesis to occur, you need light. The correct answer is option C, anaerobic sulfate reducing bacteria. So I told you the bottommost layer of the Vinogradsky column, it is anaerobic. Uh, and we have sulfate reducing bacteria that can reduce the sulfates and sulfides to hydrogen sulfide, which will be released as hydrogen sulfide is gaseous. It will be released and it will reach the top of the column, and then organisms that can use hydrogen sulfide can grow at the top, at the upper layer of the column. Okay. So this was the last question of the session. Anyone has any doubt in any of the questions? I hope everything was clear. Okay. If there's no doubt, I can end this session now. Thank you everyone for joining. I'll close the session.